Doke. So I've been given the green light to begin. If everybody can take a seat. When I began doing pharmacokinetic trials in the U.S., I met with Don Stansky, who is just extraordinary as far as his models of pharmacokinetic modeling. But he was getting busy, and he asked me if I would work with his young resident, who is it? Stephen Schaefer. And they were working at that time on a 3-compartment model for describing the pharmacokinetics very adequately for drugs in healthy individuals. But today, what has taken over pharmacokinetic modeling is a physiological-based pharmacokinetic modeling, which requires us to understand drugs in various types of individuals who have different conditions. But today I'm just going to focus on what happens in individuals when levels of blood lipids vary, when they go up and when they go down. How does that influence propofol? And then I'll tell you how it's going to influence almost all of the anesthetics that we work with. So it's important to understand that propofol is a yellow oil. The package insert is wrong. It says slightly soluble in water. It's not soluble. It's immiscible in water, and that's been shown at the National Library of Medicine, Pub Chemistry. It says insoluble in water. The other thing about lipophilic drugs that you need to know is they are extremely potent. But lipophilic anesthetics have a very long duration of action. The classic one is methoxyflurane that had a very long recovery period. But guess what? Propofol was formulated in a lipid emulsion formulation. And what happens in a lipid emulsion formulation is the plasma is 92% water, right? Propofol does not want to be there. But you put it in an oil droplet, intralipid soybean oil droplet, and it's happy until it gets to the brain and it sees lipid and it goes sucks right into the brain and puts the patient to sleep. As the lipid circulates through the body, it gets scrubbed or cleaned of all the propofol, and when it returns to the brain, it's mainly lipid, and the propofol is pulled out. And that's what creates the short duration of action, is the lipid. It's like a lipid bolus rescue from the anesthesia. This has been shown in both human and animal models, that the lipid is much shorter acting than the water version of propofol. But understanding that this physical chemistry of an oil and water is very useful because it also helps us understand another situation, and that is where and this is rare, it's not common. In fact, there's more papers showing tolerance doesn't develop with propofol. But in a few patients, you see escalating doses of the induction dose and the maintenance dose. Why is this? This is because this person doesn't clear lipids as well. And so as you give more and more propofol, you're getting more and more lipid, and they're building up the lipids in their blood system. And so as there's more lipids in the blood, it takes a higher dose for the propofol to reach the brain. So you get a decrease. You have increase in dosage, and you have a decrease in the efficacy of propofol because the lipid prevents the propofol from leaving the blood and distributing to the brain. We can also see this with other anesthetics. You can see it with the anesthetics and the analgesics, the neuromuscular blocking agents, but you can also see it, there's been at least 3 reports of warfarin-induced tolerance under hyperlipidemia in the blood. I'm going to allow these slides to be posted on the APSF website, so you will be able to get all of this information after the talk. Here's the back. There's the inhalations, agents, and which ones are water-soluble versus which ones are lipid-soluble. Same for the analgesics. So if you have a person with hyperlipidemia, one's going to be comfortable, one's not. You need to think about that as they're leaving if you've given them a big dose of Propofol. And this has been reported before, and here's the neuromuscular blocking agents. I have not found this in the literature yet, but I've seen it for Forane, I've seen it for fentanyl being ineffective in patients that are hyperlipidemic. Okay, something that occurred since I retired in 2009 is somebody put into the package insert that Propofol is contraindicated in patients who are egg and soy sensitive. This is malarkey. Intralipid, which has been around almost twice as long as Propofol, never had this warning. They actually have higher concentrations of soybean oil and the soy oil and the egg phospholipids. There's been a major publication recently come out. There's no evidence for contraindications to the use of Propofol in patients that are allergic to egg, soy, or peanut. They went back and looked at every case of sensitivity reported, and nobody had done the second challenge or identified any antibodies in the thing. I also had patients in the clinical database at AstraZeneca who had egg allergies and never reacted. This needs to come out. In 2009, when I retired, Steve Schaefer asked me to write a review for Propofol infusion syndrome so that people could better understand the syndrome. At that time, I had an inkling, and now I have 5 lines of scientific evidence corroborating what then was just a feeling, and now it's pretty firm. So let me first debunk some of the myths that are out there with regard to Propofol. It is not toxic to mitochondria. The studies have been tissue in water-based buffers, in vitro studies. What happens when you put an oil in the water? It goes right to the tissue. Or the mitochondrial, so they're valid, but only if you multiply the concentrations by 6,761. Then you get the relevant concentrations. So studies in isolated mitochondria or even in tissues are not correct. And we also have studies that have been done for cardiac patients. I apologize, this is a little bit off the slide, but these have been trumped by a study that I did in the early 90s with Richard Hall, in which patients had low ejection fractions, less than 50%, greater than 25%. There was no evidence of myocardial ischemia with Propofol. That actually reduced myocardial lactate levels versus controls, and there was less hypotension than previously seen in other cardiac studies. He thought when he started the study his heart wasn't going to last. He says, I don't know what the action is going to be on the patients, but I'm not sure I can take this. He thought he saw less hypotension because they were mostly in congestive failure, so they were volume full from a venous point of view. If you want the summary of what Propofol does in the cardiac system, It's a great sympatholytic agent that can be a patient benefit or a detriment, depending upon how you use it and how you understand the patient's sympathetic system when they're coming in. There is no similarity between CoQ10 and Propofol. I don't know who ever proposed that. Ridiculous. Okay, mitochondrial disease is what I'm going to propose to you, that I have 5 scientific lines of evidence linking propofol infusion syndrome with mitochondrial disease. In fairness to the authors who reported propofol infusion syndrome, very little was known in the 90s about mitochondrial disease. But what we do know is patients that come in they describe it as like trying to run a whole household on a little battery. Their system for converting fuels into energy is broken. This is the situation with mitochondria. They inherit, They inherit mitochondrial DNA from the mother's primordial germ cell, but they can do it at different levels. So you can have a high level of mutation where most of the mitochondria have the disease, or an intermediate level, or a low level. I'm sure most of us have some sort of level of mitochondrial disease at the present time. Look how many diseases, mitochondrial diseases, have been identified so far. And if you just limit it to fatty acid oxidation diseases, it's still a lot. And so what I want you to do is think about feeding your mitochondria when they come in for anesthesia, okay? I'm being a little bit graphic here. So what are the fuel sources? There's 3 fuel sources: glucose, fatty acids, and amino acids. So our propofol emulsion administers what proportion of those? Does anybody know? Only one of them. And you need the glucose, you need the carbohydrates to utilize the fats. You can't just live on fatty acids. The mitochondrial machine will break down. So what does this tell us? And the other thing you need to know about mitochondrial disease patients, at baseline, before they're in the hospital, they have an inadequate nutritional intake, and this has been shown here. If you look at the intralipid package insert, I didn't include this statement in the propofol label that 60% of the total caloric input to the patient should be the fatty acids. You should be supplementing with another energy source. Lactated ringers is not an energy source. Saline's not an energy source. Let me do 101 nutrition here. Dextrose is the key to giving these patients an alternate fuel source. Do not give lactated ringers, because lactic acid is a common toxic byproduct found in 80% of patients with or without symptoms as they're coming into the hospital. Heart, skeletal muscle, kidneys all prefer fatty acids. Okay, so it's no surprise that we're seeing cardiomyopathies, rhabdomyolysis, liver and kidney dysfunction. The brain uses glucose. It doesn't prefer fatty acids. It prefers glucose. There's only one cell in the body, type of cell in the body that doesn't have mitochondria: red blood cell. Okay, so here are the 5 scientific links of PRIS to mitochondrial disease. I'm going to have to cut off this talk because I'm just running out of time. But I reviewed all of the case reports, propofol infusion case reports, the first 50, so there were adults and there were pediatric, nobody talked about what they were giving for fuel. Nobody talked about the length of the NPO period. They didn't talk about what they were giving any dextrose. So this was a blind side for all of the people who were dealing with propofol infusion syndrome. Now we see that there are definite biochemical links. There's 4 separate references that have seen elevated acetylcarnitine derivatives. This is classic mitochondrial disease. There's genetic links to mitochondrial disease in PRIS patients. And now there's even a paper on ultrastructural link to mitochondrial disease. And those are all the references, so I incorporated it. So here you see recommendations from one mitochondrial disease organization: avoid lactated ringers, try to schedule the patients as early in the morning to minimize NPO time. You can even bring them in and start the dextrose as you're starting NPO. And some of the sources even recommend don't start 5%, that's not enough, start 10%. And there's even, in my talk later on today, I'll suggest a formulation of propofol that could avoid any crises in the mitochondrial disease patients. So just, you can see the rest of my slides. Please go. I've put in a whole bunch of links. Those are websites for various mitochondrial disease organizations. So thank you very much. So I welcome to the stage our next speaker, who is going to talk to us, Jeff Feldman, about the safe and effective use of CO2 absorbents. And please adjust the level of this.
Thank you. Thank you, Dave. Yeah, good morning. So we're going to transition from intravenous anesthesia to inhalation anesthetics. And when you guys are ready, put the slides up. I've titled the talk Safe and Effective Use of CO2 Absorbents because there are safety concerns that have been raised historically about CO2 absorbents that have influenced our clinical practice. And those clinical practices still persist at a time when probably they don't need to. And what I'll try to show is they get in the way of the effective use of CO2 absorbents. I hate to use the word cliffhanger in a talk on CO2 absorbents. It seems a little bit inappropriate. But to Mark's point about implementation, I do want to provide some recommendations at the end, and one in particular that I think still requires implementation in order to get to the really effective use of CO2 absorbents in our practice. I have a couple disclosures. There's a couple of companies, Clearline MD and Draeger Medical. I've done some consulting in the last year or so. So I know there's people in the audience that recognize this machine. This was the Siemens 900C ICU ventilator. And on one side of this one, you'll notice there's an isoflurane vaporizer. So a lot of us really liked this ventilator at the time. It was much better than the anesthesia machines, and we wanted to anesthetize our patients with it. So we hung the company put an isoflurane vaporizer on it so we could provide anesthesia. And in a lot of ways, an ICU ventilator is a really appealing device to anesthetize patients with. The circuit is really simple. There's an inspiratory and an expiratory limb. And yet, the machine we just saw is now really of historical significance only. And what we've done is we've really increased the ventilator capacities of our anesthesia machines. So why do we do this? Why do we persist with these machines that are more complex than the ICU ventilators? And introduce a lot of challenges even to understanding how to use them well. And it's not so that we can deliver anesthetics. We were able to do that with an ICU ventilator. It's so we're able to deliver the anesthetics efficiently, and in particular, provide a path for recycling exhaled anesthetics back to the patient. It's the only reason that we use an anesthesia machine rather than an ICU ventilator. In order to do that safely, requires CO2 absorbents to remove the carbon dioxide. So are CO2 absorbents safe? Well, historically, there's been 3 main areas of safety concerns that have been raised. The first one happened shortly after sevoflurane was introduced. There were fires, exploding CO2 canisters in the operating room. And we figured what that one was. The sevoflurane interacted with dried barrel lime. We got rid of barrel lime, and that problem went away. So we don't have to worry about that anymore. Compound A production is still something that we talk about. It happens in the presence of sevoflurane. There needs to be a strong base, sodium hydroxide, potassium hydroxide, in combination with the calcium hydroxide in order for compound A to be produced. The clinical relevance is very questionable, a lot of controversy over it. Yet our clinical practices are still influenced by the minimum flow recommendations around sevoflurane. Carbon monoxide production is the other safety concern that's historically been raised. Desflurane is the worst offender, then isoflurane, then sevoflurane. All will produce carbon monoxide. You need a dried absorbent in order for that to occur, and also the presence of strong base. And we have clinical practices that persist to today that are intended to prevent desiccation of absorbents. And again, to get to the effectiveness point, those clinical practices get in the way of the effective use of the anesthesia machine, as I'll try to show. So the APSF was on this early. In 1994, there was an article in the newsletter identifying patients who had experienced carbon monoxide poisoning in the operating room. And what's interesting about the title is it says the relation to halogenated agents is still not clear. So case reports, we really didn't know what was going on. Fast forward 11 years, and the APSF convened a conference like this one on carbon dioxide absorbent desiccation. And there were a number of recommendations that came out of that conference. So clearly identified that absorbent desiccation produced carbon monoxide, recommended that fresh gas flows be turned off between cases, that we change absorbent regularly. For example, on Monday morning, many practices adopted that approach. Whatever was the case, they would just change all the absorbents, whether it was exhausted or not. Changing when the color indicates exhaustion. And it was clear that the presence of the strong base increased both the carbon monoxide and the compound A. Potassium hydroxide was the worst. Sodium hydroxide was a contributor. And interestingly, there was a recommendation to create an expert task force to define further the characteristics of carbon dioxide absorbents that do not significantly degrade volatile anesthetics. From what I could tell, that expert task force was never really convened. I think all the data existed at that time to answer these questions, but I'll show a little of that data to make the point. So what have we learned since then? Well, the absorbent formulations have changed since certainly the 90s and even the early 2000s. The goal of changing the formulations was to minimize or eliminate the interaction with the anesthetics. So there would no longer be degradation to carbon monoxide and compound A, but also maximize the absorptive capacity. And as you'll see, there's a trade-off between minimizing the strong base and maximizing the absorption. So the manufacturers have reduced or eliminated the strong base catalyst and tried to develop alternate chemistry catalysts that didn't have the same interactions. There's also been some technology changes, and that'll be part of my last sort of cliffhanger, and it's related to the way that the canisters have been designed, which really will help us to use them more effectively if we take advantage of it. But there is a safety concern there. So just to give you some background information on how this evolution has occurred, this is an in vitro study of formulations of CO2 absorbent that were prepared just for the study. They weren't commercially available. And there were varying concentrations of potassium hydroxide and sodium hydroxide. And as you can see, that the average carbon monoxide produced was much higher in the presence of potassium hydroxide than sodium hydroxide. And as you reduce the concentrations of both those bases, the carbon monoxide production went down. And you may not be able to see, but there's actually a data point down very close to the origin, and that's calcium hydroxide alone, without any strong base, produced basically no carbon monoxide. The other part of that study, though, was to look at the absorptive capacity by looking at when CO2 appeared in the inspiratory gas. So when the CO2 appeared in the effluent from the absorbent. And you can see that with calcium hydroxide alone, it appeared much sooner than in the presence of strong base. So the strong base increased the absorptive capacity, but at the same time, you had the potential for carbon monoxide production. This is a follow-up study looking at commercially available absorbents at the time. It was done in 2000. And the point I want to make here is that if you look at, and all these studies, by the way, are in desiccated absorbents, the data that I'm showing you. In this case, the calcium hydroxide and lithium hydroxide alone at the very bottom of the carbon monoxide and compound A plots showed no carbon monoxide production, very little compound A production. And as soon as you accumulated various degrees of strong base, the concentrations of both of those agents increased. This is the only study I was able to find that actually had in vivo data. Everything else was done in the lab with just samples of absorbents and test tubes. And so this was a pig model and looking at, again, commercially available absorbents with varying degrees of strong base. And at the top plot, you see desflurane and isoflurane and carbon monoxide concentrations that were detected. Note that the scale for carbon monoxide is very different in desflurane and isoflurane. Isoflurane is much lower concentrations of carbon monoxide. But again, you look at the product new soda lime, which had 2.6% sodium hydroxide, no potassium, and it's very close to the calcium hydroxide alone. So a small amount of strong base catalyst increased the carbon monoxide a little bit, but not much. And if you look at the carboxyhemoglobin levels in the pigs, you can see there's just a little bit of increase from baseline in the presence of the strong base compared to calcium hydroxide with no strong base. This is some data on just sevoflurane and compound A, and it was just a nice contrast of hydrated versus desiccated absorbents and carbon monoxide and compound A production. And the absorbents at the top all contained sodium hydroxide. Ones at the bottom had no sodium hydroxide. And you can see that in the absence of any sodium hydroxide, there's no carbon monoxide production at all, fresh or dried. And the compound A production also goes away, although if you dry the Absorb, which is calcium hydroxide only, and lithium hydroxide, you get a little bit of compound A production. So to try to hone in on this, these are probably the best two sets of data I could come up with to really present today, where they compared Dragersorb Free, which has a small amount of sodium hydroxide, and Absorb Plus, which is sodium hydroxide free. And there's two studies here. The STRI study, both were in vitro. The STRI study used fresh gas flow of 500 mLs per minute, and they found that compound A was less than 1 part per million with both absorbents, and they stated that no carbon monoxide was detected with desiccated desflurane. But if you look at the paper, they really didn't present much of the data, so I'm not quite sure exactly what to make of that. Then in another study, Kobayashi studied just sevoflurane, and this is the data here looking at compound A production. And he compared those 4 absorbents on the right. The Sodazorb had sodium hydroxide and potassium hydroxide, produced some compound A, and all the other 3 had either very low or no sodium hydroxide, and you can see there was very little compound A production, very little difference between them. And the absorptive capacity, when you looked at time to appearance of CO2, was a little bit better for the Dragersorb Free with the sodium hydroxide, and the Absorb Plus was pretty close, and they were not much different from the Sodazorb that had a lot more strong base. So the message is you can reduce the strong base, almost eliminate, if not completely eliminate, the compound A and carbon monoxide production, still maintain pretty good absorptive capacity. So not all the data is completely historic. This is data from Jan Hendricks. It's actually in review now, but he agreed to let me show some of the data. And what he's looked at in particular is the duration of effect of the absorbents, or their absorptive capacity, depending on the amount of strong base in the formulation. And you can see that as the sodium hydroxide concentration increases, the time until inspired CO2 begins to appear in the effluent from the absorbent goes up. So you get more time per 100 grams of absorbent until CO2 appears as the strong base increases, with the exception of the Spirolith, which has a very low sodium hydroxide, but a very different geometry than all the others, and that maintains a much higher absorptive capacity at a lower sodium hydroxide concentration. So in terms of safe absorbent use, if you don't worry about desiccation, you're not going to get any carbon monoxide. If you never use sevoflurane, you're not going to get any compound A. That said, I think all of us have practices where gas flows may be left on the machine, and there may be a concern of desiccation, or certainly sevoflurane is commonly used. But the selection of the absorbent based on what's available today can really eliminate these concerns. You can choose to minimize or eliminate the strong base, and those options are readily available for modern practice. And the absorbent capacity is going to depend to some degree upon the presence of a strong base. But the next part of my point is that these modern absorbents really do support a more effective practice and really using the anesthesia machine to good effect by maximizing the return of exhaled gases to the patient. So in terms of effective absorbent use, what you're really interested in is how efficient is the absorbent that you want to buy or put into your machines. How much CO2 is it going to absorb for the dollars that you spend? And it turns out it's really hard to make a clinical comparison. There's a lot of cost differences. The way you change the absorbent will influence the comparison, what fresh gas flow you use. I work in a pediatric hospital. Those patients, a lot of my patients produce very little CO2. So if I compare absorbent use to little patients versus big patients, I'm going to see big differences. But the efficiency, and this is really the key point I want to make today, is going to be driven by when you change the absorbent. So if you change it on a schedule, irrespective of the indicator, you have no idea how much absorbent you're wasting, throwing into the garbage. So that's probably the least efficient or least effective practice. If you wait for the indicator change, it's certainly a safe practice, but again, you still don't know how much absorbent you're throwing out. Now capnography is a standard in our machine, so we can use inspired CO2 as our indicator of when the absorbent is wasted, and that now becomes a much more effective way to use the anesthesia machine and use the absorbent that you've purchased. And fortunately, the manufacturers have provided us the ability to do that. So this is a picture of a couple of different canisters that are designed to be changed in the middle of the case without creating an open circuit or without creating a leak in the anesthesia machine. So you can open it up, take the canister out, put a new one in, close it up, and continue on. And actually, let's see. Let me show the next slide first. And we've actually adopted that practice at our hospital. We started using a lithium hydroxide absorbent a few years ago, which unfortunately is no longer available for cost concerns, but it didn't have an indicator. And so we had to go to capnography as our way to change the absorbent. And so if you look at these two displays, you can see on the left, you have a relatively low fresh gas flow. I think the total's about 500 or 600 cc's. And you can see on the capnogram at the top that there's an inspiratory CO2. I think it's about 10 millimeters of mercury. And what we do is before we change the absorbent, we increase the fresh gas flows to open the circuit. If the inspiratory CO2 goes down, we know it's the absorbent. We change the absorbent canister, return back to low flows. Works pretty well. We've been using it for a while. However, there's a gotcha. And this was actually published, I think, in 2013 in the APSF newsletter, which was a description of changing an absorber in the middle of a case and not detecting a leak. So the absorbent canister had a crack in it. During mechanical ventilation, because of the configuration of the anesthesia machine, the leak was not apparent. There's a check valve between the ventilator and the canister. And so when the ventilator would cycle to positive pressure, it would hold that valve closed. None of the inspired gas would leak in the canister. When they went to manual ventilation at the end of the case, started squeezing the bag, all of a sudden the canister leak became very apparent, and so led to kind of an exciting moment at the end of the case. So although changing the canister mid-case based on inspiratory CO2 is the most effective and efficient way to use the absorbents, we do have a safety concern in that regard. So, and talking about changing it not based on an indicator anymore and based just on inspiratory CO2, I recognize it's a little bit controversial perhaps. And so is it safe? So there may be some special populations, neurosurgery patients, patients with pulmonary hypertension where you really want to control CO2. We do monitor CO2 continuously in the operating room, and it's the arterial CO2 that's the most important. And you can correct you can maintain a normal arterial CO2, even with inspiratory CO2, by changing the minute ventilation. You can also increase the fresh gas flow and get rid of the inspiratory CO2 if you have a concern for the patient. Circuit leaks is, I think, the biggest concern here, and this is my cliffhanger, if you will. Basically, by changing the absorbent canister in the middle of the case, you're bypassing the leak test. And I've actually written, and a number of other people have written, that you should do the leak test with all the components of the machine intact. And so we're really bypassing that safety feature. And if you're going to do that in today's world, you need to be vigilant to detect leaks. You can always, if you're concerned about the canister, disconnect it, run the fresh gas flows up, and you don't have to worry about it. But it does require more vigilance, and if we get back to May's talk about kind of robust approaches to prevent problems, that's probably not the most robust approach. And what we really need is a device or a process for testing the canisters independently for leaks before they're replaced. So we get them in boxes from the manufacturer, they're shipped. We had this problem in our place with 2 canisters where a box had been dropped, canisters were cracked, changed mid-case, and we had leaks that weren't detected. So this is the thing that I think needs to be implemented in order to get to this more effective practice. So in terms of where we are today, the choice of absorbent, I think we have lots of good choices now. Would recommend something without potassium hydroxide. Small amount of sodium hydroxide seems to be safe, or if you're at all concerned, you can go with a sodium hydroxide-free absorbent. You will give up some absorptive capacity when you do that. And the pharmacoeconomics of that can be a little complex to work out. And depends really on your purchasing and what's going to cost you. I think it is beneficial to not waste your fresh gas flow, turn it off to be between cases. The machines, many of them now remind you to turn off the fresh gas flow, so they do help us. And low flow techniques, which is kind of what's embedded in this talk of using the machine more effectively, actually prevent desiccation because it maintains a lot more hydration in the absorbents. And so I would recommend that you use the anesthesia machine effectively and think about changing on inspiratory CO2, but my caveat is let's all try to get something implemented where we can really test those canisters before we change them for leaks. Thank you for your attention. I appreciate it.
We are doing great. We have one more speaker, Rayford Brown. He is talking to us about FDA and generic drug labeling updates. I met with the anesthesia advisory panel that he chairs a number of times. They are excellent in enforcing drug safety in anesthesia. So my hat's off to Rayford and his committee. You can adjust this.
I wish I could say he was right. I'm Ray Brown. You might say the topic that I have here could really be what I did on my summer vacation for the last 4 years, but it was not only my summer vacation, my winter vacation, weekends, nights, and all other times. If Kathy asks you, my wife, where I've been, please tell her that I've been working with the FDA. There you go. Thank you, sir. What we want to talk about this morning is I'm going to shift gears and talk about not about individual drugs, but about process. It is important for the FDA to be seen as doing right and to be doing the right thing all the time. And in many cases, I have taken it as my job to assure the public that the FDA, at least as it relates to anesthetic and analgesic drugs, does a little bit better job of getting it right. I don't have any financial conflicts, and I'm not going to be discussing the off-label use of drugs. I have to tell you that the opinions expressed here are not those of the FDA or the Department of Health and Human Services or the American Academy of Pediatrics, who I also work for. On the objectives that I have for us today is that I want to really get into some detail about the depth and breadth of the responsibilities of the FDA as it relates to anesthesia. I want to especially focus on areas of responsibility that are important to the practice of anesthesiology and suggest some of the current problems that may affect the future practice. I want you to think about all that you do every day. Everything that you use, from monitors to absorbents to adsorbents to tape to endotracheal tubes to all the drugs that we use. The FDA regulates all of it, all the monitors, all the drugs, even the apps that we use on our cell phones. In many ways, the FDA, I think, is the most important government entity for our profession. Some people would argue that CMS, the Center for Medicare and Medicaid Services, is, but I think that on the front end, at least, this is, the FDA is really over their heads. It's really important that they get it right all the time. They control about 25 or regulate about 25% of the gross domestic product, our gross domestic product. Now about $18 trillion, so about $4.5 trillion is in the hands of the FDA. All the drugs made in this country, but not just those drugs, the drugs that are made outside of the country, and there are lots of those. All the physiologic monitors and all the biologic products, of which there will be many more in the coming days. Now, there's a problem with this in that in order for you to be able to assess how effective an agency is, or a group of people that are regulating something as large as $4.5 trillion, you have to know what it is that they're doing. You have to be able to look inside of their decision-making process and assure that every single time it is rational and that as a consequence of the decision-making process, that the right thing will always be done. We know historically that is not true, because many of the decisions that the agency makes are confidential. In fact, most of the decisions that they make are confidential. And they're confidential to me, and they're confidential to you, and they're confidential to everybody except the people that are in the room. As they said in Hamilton, these are the people that are in the room. There's nobody else. And we don't know what their thought processes are for decision making. It's obscure, and sometimes it's never understood. An example of this is Zohydro. I'll bring up Zohydro because I've been working on opioids for the last 3.5 years. Zohydro is a hydrocodone formulation that was put out in 2012. It's a immediate release, and at the same time that the FDA agreed that could be marketed, they were telling everyone that every opioid needed to be in the form of an abuse deterrent formulation. So on the one hand, you're making a decision where you're allowing Zohydro to come on the market. On the other hand, you're telling all manufacturers that henceforth, that all of these will need to have abuse deterrent formulations. I can tell you Zohydro has none. It has no abuse deterrent formulations. And we can argue all day about whether the abuse deterrent formulations have been effective. Many believe that they have not. But This is just an example of how we can't understand from looking on the inside what's happening in the minds of the people that are making really, really important decisions for us. There are pressures on the FDA, certainly, and one of the reasons that there's such secrecy, then they will call it confidentiality, are the pressures that you see here. So there's pressures on the left-hand side of the screen that relate to the regulatory focus, such things as congressional oversight, and there is a lot. The person that I work with, Sharon Hertz, who's in charge of the division which evaluates anesthetic drugs, said that within 20 minutes of her being named as the head of the division, she got a call from a congressman wanting to know why the FDA had approved a drug which had killed a kid in his area of population. So there's a lot of congressional pressure. There's pressure for public safety. There's litigation risks that I can tell you the agency is very involved in. There's immense pressure from industry to turn out drugs at a more and more rapid pace. The FDA does many things that are not correct, but if you look at the FDA against the Europeans and the Canadians and every other well-recognized group in the world, we turn out regulatory process faster than anybody else in the world. And it may help you to know that we're spending $5.4 billion a year, which is the budget of the FDA, to assure that that continues. But there's still pressure to turn out more and more things faster. There's historical precedent. In other words, what's the evaluation of this drug? How is it going to relate to all the other drugs? And Zohydro, again, is kind of an example of that, because Zohydro was evaluated after the agency had said we need to have ADF formulations. And then there's patient advocacy pressure, and it is immense. To some extent, patient advocacy pressure can be tied to industry because industry pays for a lot of patient advocacy. As I said, I've spent the last 3 or 4 years hearing stories about people that feared that they wouldn't be able to get all the opioids that are required. And I just have to say that industry pays for those people to come to Washington to tell us about that. I'm not suggesting by any stretch that that is correct or incorrect, but the two things can occur together. Now, the regulatory process on the other side of the slide has implications for what happens in Congress, for industry and how they view the drugs that they're going to look at, and for patient advocacy, and of course for the historical element. The pressures on the FDA don't stop with those, however, because the pressures that the FDA is feeling now relate in many regards to bad science and lack of data. And we heard a lot about that this morning, about the lack of data about nitrous oxide. Well, who would have thought that after 1 million years of using nitrous oxide, that we wouldn't have all of the data that is required to determine whether or not it is indeed in a given patient safe to use nitrous oxide? But we don't. And what kind of science can we trust anymore? When I open up any reputable journal, I don't know if you do the same, but I ask myself, can I believe what's on the printed page? And you probably, if you see my name up there, you say, this guy's nuts. This guy's nuts. I don't believe any of it. And that would be reasonable. They have other pressures from organized medicine. Organized medicine fears interference in medical practice, and that's reasonable. And their other pressure is their perceived lack of statutory authority. They don't believe that they have the wherewithal to be able to do the work that needs to be done. Now, there's some critical issues that I'm just going to mention which relate specifically to anesthesiology. Before I put any of these up, I want to say I'm not here to discuss the science. Can we agree on that? I'm not here to discuss the science. I'm here to talk about the process and to ask questions that I want you to carry with you. Anesthetic neurotoxicity is a large part of the work that pediatric anesthesiologists and others have been trying to get a handle on for the last several years. It is important because we know and have known for some time now that general anesthetic agents produce in laboratory animals accelerated neuronal apoptosis. Seems to be associated with developmental delay. There's some suggestion of some effects in humans. But why are we just doing that now? The best I can determine, halothane was marketed before, well, I have to carry it with me then. I heard somebody say this morning, I'm a walker. Well, I'm a walker too. Why are we just figuring this out now when at the very least we've known that this was a problem with agents like general anesthetic agents since 1985 when Olney produced his original research in St. Louis? Why are we just figuring that out at this point? Part of the reason is that many of these agents are not tested prior to marketing in models of the developing central nervous system. We have a circumstance where we think we're doing the right thing, but we're not doing the right thing. We're not testing these agents in every single population where they will be used. And one of the revealing things to me has been that we depend on post-market testing programs. Thanks, Dad. Can you hear me now? We depend on post-market testing programs, which have not been done for 30 years in some of the agents, such as general anesthetic agents, because we need to know in large populations, millions of patients, what it is, what are the characteristics and the behaviors of these agents after they are seen in multiple populations. When agents are tested, when sevoflurane was tested, it wasn't tested in developmental. The developmental aspects of this were not tested. Before we began to look at it 30 years later, these were not brought to the fore. So I finally got to point one. Point one is that the testing of all drugs marketed for human consumption must be complete and must reflect all of the clinical conditions under which the drug will be used. That is currently not the case. Followed up by point two, the pre-market evaluation of every drug that passes through the FDA is incomplete. Why is it incomplete? It's incomplete because there is a minimum of testing by the sponsors. They're not, and that's fair. They're only doing the minimum because it costs them money to do this testing. If we want more testing, we have to ask for more testing. So we have to have, and we depend on, post-market evaluation of these drugs in order to determine in large populations what the toxicities are. And unfortunately, the FDA over the last 25 years has not been very good about enforcing post-marketing evaluations. So, number 2, opioid regulation. Opioid regulation is a major role of the agency. It approves the labeling of compounds which can drive clinicians to use an opioid for a particular process, such as chronic pain. If you look at all the long-acting opioids now, you will see that they're approved for using in chronic pain. Unfortunately, there's no data, scientific data, to support that. Now, there's plenty of epidemiologic data to support that some people get better on it, but there's no scientific data which support that. But the FDA has said that it's approved for the use in chronic pain, and that, ladies and gentlemen, makes it all right. They're capable of creating new formulations by requiring industry to respond, and they approved and pushed industry to determine that abuse deterrent formulations were useful. And so Ron and I have spent the last years of our life evaluating ADF after ADF after ADF. We're not certain that any of the decisions that we make about any of these drugs are the correct decisions because we don't know what goes on behind closed doors. And the organizational structure doesn't really consider secondary consequences of opaque decision-making processes. In the case of ADFs, abuse deterrent formulations, there was a monstrous rise in the use of heroin after the development and licensing of ADFs. Now, did we cause that, or was that, what caused that? We're not certain, but it looks like that by taking drugs away from people that were going to use them anyway, that we increased the number of patients that were going to be transferred or would transfer themselves to the use of heroin, fentanyl, and other illicits. Point 3, there's no system of governance where secrecy is a defining characteristic that can be expected to provide consistent, rational answers to difficult questions. The insular nature of large government organizations focuses on protecting past mistakes from future review and changing in the method of analysis in the near term. Those are harsh words, and you can believe them or not, and we can argue about them later. But after 4 years of examining this agency, I think that questions need to be asked about this. I want to talk for just a second about generic medications and the labeling of generics, because it's important to our practice. How do we know that a generic is as good as the agents that we otherwise use? We know because there are definitions that the FDA enforces. And the standards include that the medicine needs to be of the same strength. It needs to have the same type of, be the same type of product, such as a tablet or an injectable. The medicine has to have the same route of administration, such as an oral or topical, and it has to have the same use indications. Now, further, and these are perhaps more important, the inactive ingredients of the medicine are need to be acceptable. And one of the things we have been looking at recently is the excipient compounds in the drugs that we use, because some of the excipient compounds are in fact quite toxic. And we have determined in at least one case of an opioid that went to generic formulation that the excipient produced pretty substantial pathology. These agents need to last for at least the same amount of time, and I'm going to get back to that. It needs to be manufactured under the same strict standards as the brand name, and the container in which the medicine will be shipped and sold is appropriate. And the label is the brand name medicine's label. Now, the last point is this. The 90% confidence intervals for the area under the curve of concentration of these drugs only needs to be within 80 to 125%. So they don't have to be the same. Generics are similar, but they're not the same. So why focus on these? Why focus on these areas today? The FDA makes decisions that affect our patients and our professions. It's important that we know that the process is rational. The safety of patients depends on the agency. We must be able to trust that they're doing the right thing. And right now, I worry about that. Thanks for listening. Talk as we sit in front of the panel.
Okay, so let's bring the panelists up. And now we're going to somehow get 6 chairs around this table, or maybe a couple people can even stand here. Five chairs. Are there 5? There are 5. Yeah. Okay, great. And when you ask your questions, either go to the mic there or let me know. I'll run a mic out to you, because this is being video recorded, so if we can get your questions on the record, that'd be great. Not trying to put you on the spot.
We have a mic right there in the middle of the aisle. The mic.
Jim Phillip, Brigham and Women's Hospital. Jeff Feldman, question on the CO2 absorber change during the case. Even if we were to test the absorber before we plugged it in, we would not know that it was secure in there, so we still wouldn't have done the full test. One other point on the CO2 absorber is the inhaled agent is missing from it, so we've got to overcome that loss. Typically 1 cc of liquid anesthetic agent is behind by.
So this is unique questions from Jim Phillip, who does quantitative anesthesia like no one else I know. And so you're absolutely correct. You have, with regard to point 2, they're going to have to build up some concentration in the circuit. And if you're trying to maintain a very low technique, you may see a dip in your concentration until you do that, clearly. And I haven't studied that. It'd be interesting to know what the... No, but in terms of looking at how the inspired concentration changes and how long it takes to restore at different fresh gas flows would be an interesting thing to look at. And then your first question was with regard to engagement of the canister and the potential for leaks. I think that's possible. We've had good success with those things, but again, like anything we do, vigilance and monitoring is going to be important for every phase of the anesthetic, and particularly when you make a change like that.
Next question.
Yes, Jeff Shapiro, Mednax, Atlanta. Ray, this question's for you. What is the explanation and rationale for the lack of transparency from the FDA? To me, that's very concerning because it just covers up a lot of decision-making, which isn't necessarily may be in the patient's best interest.
Well, I appreciate.
Does this need this mic?
No. I appreciate your question, and you'll be interested to know that that the FDA has formed a transparency expert work group 3 or 4 years ago to look at being more transparent. I think you're absolutely right. It's likely never in the best interest of patients or physicians for us not to know what the decision-making process is. To be eliminated for that asks for bad things to happen, and there is no explanation. If the observation is economic, when the advisory committee makes a finding on a Thursday about a drug, an individual drug, an organization can go out of business on Friday. And that has happened. I mean, we've made a finding. Of course, it was all Ron's work. We've made a finding on one day, and the next day, the stock dropped by 100%. So that's the only thought that has been put to me.
I'm concerned about the influence on that decision making.
The additional question was he was concerned about the decision-making process. The influences, yeah.
Absolutely. That's the reason I'm bringing it to the Anesthesia Patient Safety Foundation, because this is a large organization which is trying to do the right thing. I've been trying to do the right thing, but dealing with a large government bureaucracy is extremely difficult. Thank you.
Next question.
Tillman Hein Dallas. I have a question or a comment to Dr. Brown, and it may be semantic. I believe that the FDA has a definition of labeling that refers to the package inserts, the printing of therapeutic claims, safety warnings, and so forth. I'm a clinical anesthesiologist. When I say labeling, I mean what Dr. Bowdle showed us a picture of, namely the ever-changing packaging and labeling of medications that arrive in my Pyxis machine or drug box. The ASA has a very nice medication labeling policy that sadly restricts that advice to anesthesiologists putting labels on syringes. I would like to see this body of people make a recommendation to extend that labeling recommendation to the pharmaceutical industry as well, so that when these medications arrive in my drug box, they have for an opioid a blue cap and for a muscle relaxant a red cap. I have to admit, just like Dr. Pion Smith admitted to a mistake just a few days ago when I drew up sufentanil into a syringe and I intended to draw up ondansetron. They had the same cap, the same print, and by the way, those prints on the medication vials, you have to use a microscope to read them. Thank you.
Thank you for your comment.
Next question. Or
Brian
comment.
Thomas with Preferred Physicians Medical. Dr. Hogan, as a MD JD, I would like to ask you a legal question with regard to informed consent and the use of nitrous oxide for OB patients. Many of our policyholders around the country are more frequently asking our advice on this very issue. Should it be something that's addressed in the informed consent process?
I'll just answer in one word, yes. I mean, there's no question. And like I said, if you ping me with your email, I'll send you our law review on informed consent and anesthesia. And there's just a host of reasons. The law differs from state to state. It's not federal law, it's state law. So there's reasonable physician standards in some states, reasonable patients in other states. It's all devolving down to the reasonable physician as a patient. What would you do for your family members? And given the fact that I don't think the FDA is going to be a route to correcting the problems we're having with nitrous oxide now. I spent 2 semesters studying FDA law, and it got very nihilistic. I'm not sure it was as nihilistic as our talk this morning. But if we wait for the FDA to do these things, it won't happen. But we have a sword and a shield, and that is what I call the Magna Carta of being a physician, and that's informed consent. And if we're giving people drugs that are not FDA approved for safety or efficacy, I think it's only fair that they be aware of that.
Thank you.
And we'll put a link on the website for that article. You could tell them.
Sure. Yeah.
And could I refute that?
Yeah.
If you're a pregnant
Wait
woman,
a second. Can you turn this on, please?
if you're a pregnant woman, heavily into labor, informed consent, you'll say yes to anything to get pain relief. So I think we need to take it a little bit farther and not just say, because our patient agreed to it, we're going to give you a taxa.
So, yes and no. I agree with your point, and it has to do with the capacity to give informed consent. And that's why I think the capacity for informed consent should be provided way before you're in labor. Okay, we're going to discuss your options for analgesia, and we'll make plans accordingly. What do you want? And some people are going to change, and we're going to have to ride the horse when they change. But this is something that can be addressed easily in labor delivery weeks or months before.
Next question.
All right, it's Jan Aaronworth from Yale University School of Medicine. Feldman, you're up. So, Jeff, I agree with what you say. I think people lose sight of the fact that carbon dioxide is much less important than we think. And if you go back to 1959, Jack Fruman published an article called Apneic Oxygenation in Man, where they had patients apneic up to 1 hour with PCO2s of 200 to 250 without any deleterious effects. So a CO2 rise of 10 or 15 millimeters of mercury, unless you have a neurosurgical patient with increased ICP, really is trivial. It doesn't make any difference. In fact, when we're doing thoracic anesthesia, we're frequently running low tidal volumes and high PCO2s in 50s, 60s, 70s, 80s. So the point is, I think this changing of CO2 canisters mid-case, should be eliminated, really. It just makes no sense. You can increase your minute ventilation. You can live with a PCO2 of 50 without any problem whatsoever. But it does introduce this whole thing that you mentioned about potential cracks, potential leaks, which is really a much bigger problem than a few millimeter rise in PCO2.
So the contrast you're making is a little different than what I was thinking about. So if I understand you, you're saying if the canister should become exhausted in the middle of the case, leave it in place and manage the CO2 in other ways, fresh gas flow, minute ventilation, whatever, until the end of the case, and not open the circuit. Certainly a reasonable approach. I think the contrast I'm trying to make is, in our current practice, a lot of it informed by our safety concerns about absorbents that are historic, we're wasting a lot of CO2 absorbent. So by changing it on a schedule or changing on an indicator, we're throwing a lot of it out. We're spending money on it, it's polluting the environment, it's just a waste. So your proposal's an interesting one, can add that to the list of how to use CO2 more effectively. Well, if it gets absorbed, gets exhausted, just run up your flows.
I agree, and the newer compounds, the lithium ones, which are much more expensive, if you keep changing them on a weekly basis or whatever, then you've got a real financial problem. And somebody from the bean counters is going to come back and say, go back to Sotazorb.
Yeah.
If we could, this will be the last 4 questions, because then we'll take a break, because I know a lot of people would like to take a break too. Thank you.
Okay, next question.
Thank you, Ron Littman. So I have a couple quick comments and then a question for you, David. The first comment is in response to the gentleman who was up here about the labeling and the colors. It is so much more complicated than people realize, and later today I will talk about it a little bit in the 10 or 12 minutes I have. And the second comment is to Ray. A lot of people really can't appreciate some of the decisions that made, and what Ray's talk reminded me about was earlier this year. You all should know about this, and it's, by the way, everything that the anesthesia committee does with the FDA is on the website word for word. Our transcripts are there, but not many people know how to get to it, but it's all publicly available. So anyway, we evaluated that, I don't even remember the name of it, but it's the long-acting local anesthetic from Pacira, and And they showed us study after study that showed that essentially it's not much better than regular bupivacaine, and it's not, in fact, some studies showed it wasn't better than placebo, and they had no safety data. And some animal studies suggest that if you inject it into a vein, you cannot resuscitate from cardiotoxicity with intralipid. The FDA has never approved a local anesthetic for a specific kind of a block. They only just approved local anesthetics, like, okay, it's approved for local anesthesia. But in this case, we voted it down, I think 6-4, but they came back with an indication for it for shoulder blocks only. I mean, that's insane. I don't know about you, Ray, but when I saw that, I was like, what? They had never, that's the first time the FDA in history had done it. So the only thing I can come up with is a cynical sort of response that the industry had some influence. Anyway, so the question I had, David, in pediatric anesthesia, we hear a lot about the use of propofol for mitochondrial patients could be bad, but it's not propofol infusion syndrome per se. It's some other whatever. So for a certain type of mitochondrial disease, that it could be bad based on some abstracts. I'm not really sure if it's published even.
Yeah, no, it's based upon in vitro studies of isolated mitochondria that are suspended in water. And then they put propofol in, and the propofol goes to the tissue like iron fragments go to a magnet. It's going to be, boom, full of propofol, because propofol hates water. So it's not going to, those are irrelevant. By the way, if you think about the implications of propofol inhibited mitochondria, every patient that you have would be having major effects, because that's the energy, that's the engine in your body that produces all the ability to live, to beat, to send nerve signals. So it makes no sense that propofol would only affect rare patients if it indeed inhibited mitochondria. So anyway.
Barry Metz, Penn State, just a quick observation. We talk about labeling and colors and different vials, etc., but what we don't talk about is the fact that most of the time we're administering these in dusky, dark environments, when the laparoscopes have gone on and the lights have gone down. And that's a huge safety issue, but that's just one component. I wanted to ask Dr. Brown a question. You said something about generic substitution, but you didn't add on your slide that it should be the same structure of the drug. Was that something you missed, or is that something
No,
I've missed? I thought it had to be the same structure of the drug for a generic substitution.
The items that were on those slides were taken directly from the FDA regulatory manual concerning generic drugs. And I may have left one out, but I don't think so. I think that I'll go back and review that. But
because wouldn't that be the essence of it? A generic substitution would be exactly the same drug formulation, but that was left off your slide.
Okay.
Yeah, just a question. That's what I always thought generic substitution was. The same drug.
Well, there is a specific definition, and it may well be that, or we may be talking about a different syntax in that they think that one thing means something, and you think that something means something. I'll go back and review it.
I think that's the essence of it. Thank you.
Can I just add a comment to that? Propofol was an AstraZeneca product until the generic was introduced. And we sued the FDA. We sued them because it wasn't an equivalent drug. It contained, instead of EDTA, it contained sodium metabisulfite. And there is published in the APSF newsletter a report of 2 frank bronchospasms because the pharmacist did not tell the anesthesiologist that he had substituted the sodium metabisulfite formulation. And so they're equivalent as far as the main ingredients, but all of the, what they call inactive ingredients, that they're generally recognized as safe. It's almost equivalent to nitrous oxide. They've been around forever, but some of them, we argued, are not safe. So, two quick questions.
Jim Suchik, Michigan Medicine. I've probably delivered about between 1 and 2 dirigibles worth of nitrous oxide, and now I'm getting a little bit scared. But when we have to make these decisions upon which drug or drug combination to use, it's always relative. Do we have any information of the frequency of these deleterious effects of nitrous oxide? Because we have previous speakers saying, well, desflurane and SIVO will kill the liver also, so I can kill your nerves this way, I can kill your liver that way. What are the numbers relating to this? Because I've given a lot of nitrous. I have not seen those particular side effects, but I don't know if I'm not looking hard enough what the incidence is of these numbers so I can make a logical, rational decision for my patient.
There's no data because we haven't looked. It's just that simple. And like I say, a lot depends on how much damage is acceptable, and that is determined by how much damage you look for. And if you don't look, you can't see.
I'll just add that the onset of the myeloneuropathy is like 2 to 3 weeks after anesthesia. You're not going to see it. It's a hidden devil. Yeah, final question.
Yeah, Tom Krejci from Northwestern in Chicago. Another follow-up on the labeling. The ASA, by way of the Equipment and Facilities Subcommittee, did propose labeling guidelines, which the ASA has published, and did take that through the House of Delegates and got it approved, took that to the FDA where it was rejected. The main reason for the rejection is that we will not deal with just anesthetic drugs or perioperative drugs. We would have to deal with all injectables, and there's not enough colors in the rainbow to cover all of the medications in the hospital. It was more extensive than that, but that was the nuts and bolts. Not saying we can't go back again, because that was over 10 years ago, but it was addressed.
Okay, so Mark, where are we at?
Tom, to that point, by the way, there are some different groups now that are starting to reconsider that anesthesia drugs could be different than other type of drugs. So we'll talk about that this afternoon. Right, Ron? Yeah. Okay, so folks, let's take a break. Let's come back at 5 after. That'll give you 25 minutes to go out and have a break, and I'll round you up the usual way. If you hear a bellow, it's me.